morning or good evening. My name is Bert Hofman, and it is my great pleasure to welcome you at this 171st Qatar Lecture. The Qatar Lecture in Preventive Medicine is the oldest institutionalized lecture in the field of epidemiology and preventive medicine. Since 1912, the lecture has been administered under the auspices of the Department of Epidemiology, now of the Harvard T.H. Chan School of Public Health, and it has been done so under the bequest of John Clarence Qatar, graduate of Harvard Medical School. I will say a few words about Dr. Qatar. Dr. John Clarence Qatar was born in Warren, Massachusetts in 1851. He graduated from Harvard Medical School in 1877. 171st Qatar Lecture will today be delivered by Dr. Elizabeth Weiderpaas. Dr. Weiderpaas is a Brazilian researcher, naturalized Swedish and Finnish citizen, and an expert in cancer epidemiology and cancer prevention. In January 2019, Dr. Weiderpaas took office as the elected director of the International Agency of Research on Cancer, ARC, a specialized cancer agency of the WHO, based in Lyon, France. The agency has a staff of and faculty of about 150, and its governance is made of 27 countries, the states, the participating states, and I think it still includes the United States of America. Dr. Weiderpaas previously served as head of the Department of Research at the Cancer Registry of Norway and of the Genetic Epidemiology Group at the Folkhälsan Research Center in Finland. She was a professor of medical epidemiology at the Karolinska Institutet in Stockholm, Sweden, and a professor of cancer epidemiology in the Arctic University of Norway. She will give the lecture, and she will allow us to ask questions or comments after the lecture. She will address them. Now, if you have a question or a comment, please feel free to enter them in the Q&A. You can find this below the Zoom, and we will then, after her lecture, address them, or she rather will address these questions. Please join me in welcoming Dr. Elizabeth Weiderpaas as our 171st Qatar Lecture. Dr. Weiderpaas.
Thank you, Professor Hofman. Dear colleagues, participants, dear friends, it's a great privilege to be invited to give this virtual Qatar Lecture of the Harvard Chan School of Public Health. I will be talking about how evaluations are conducted for the IARC monographs on the identification of carcinogenic hazards to humans. Next slide, please. I have no conflicts of interest to disclose, and I do not work in the program that conducts the monographs. Next slide, please. So first, let me start with a brief update on the global burden and the projections for the next decades. Cancer will become the leading cause of premature death in most countries in the 21st century. The cancer incidence and mortality burden will double by 2040 from the incidence 18.1 million that was in 2018. It will reach almost 30 million by 2040. The burden of cancer will particularly affect low and middle income countries. These statements are based on IARC's cancer surveillance work that brings together cancer statistics globally. This is due not only to the demographic changes, but also to a transition in risk factors leading to a change in the pattern of cancers from those linked predominantly to infections to those linked to environmental factors, including obesity. Furthermore, it's predicted that the future increases in cancer incidence will disproportionately affect low and middle income countries, representing a significant health, social, and economic burden. The identification of environmental carcinogenic hazards to humans is more important than ever. In the right side of the figure, you can see the targets for the Sustainable Development Goals, the probability of premature death between ages 30 and 69 from cancer, and the spheres represent the target, and the dotted lines the speed of the decrease to reach these goals and we see that low and middle income countries are lagging behind developing countries in reaching them. Next slide, please. So let me briefly introduce the IARC monographs program. The IARC monographs are a series of scientific reviews that identify environmental risk factors that can increase the risk of cancer. Sometimes they are called the World Health Organization Encyclopedia of Carcinogens. The IARC monographs have reviewed more than 1,000 agents and have identified almost 500 known, probable, or possible carcinogens. The IARC monographs embody principles of scientific rigor, impartial evaluation, transparency, and consistency. For each evaluation, IARC convenes an international interdisciplinary working group of expert scientists to systematically review the pertinent scientific literature and to develop consensus evaluations and classifications. IARC selects these experts based on their knowledge and experience and in the absence of real or apparent conflicts of interest. The IARC handbooks complement the IARC monographs evaluation of carcinogenic hazards, providing evidence, synthesis, and evaluation of cancer preventive effects of chemopreventive agents and on the primary interventions and cancer screening, using the same rigorous evaluation process as the IARC monographs. The national and international health agencies can then take action to prevent avoidable exposures to known, probable, and possible carcinogens and to implement cancer preventive strategies. Individuals, too, can use this information to make better choices that will reduce their cancer risk. Next slide, please. So let's go now into detail for the monograph approach of what is new based in the publication of the preamble. When describing the monographs program, we need to start with the definition of hazard and risk. Learning the difference between them is also necessary to understand the goal of the monographs program, which is a process of cancer hazard identification. It's not a cancer risk process. Next slide, please. So what exactly do we mean? A carcinogenic hazard is an agent that has been shown to be capable of causing cancer under some exposure levels or conditions. A carcinogenic risk is an estimate of the probability or chance that cancer will occur given some level and condition of exposure to a cancer hazard. For example, hazard identification determines that tobacco smoking is a carcinogen to human, a group 1 carcinogenic, and it causes lung cancer and many other cancers. A risk assessment determines the probability or chance of developing lung cancer or other cancers associated with a given level or pattern of exposure. As another example, the hazard identification in the monographs has determined that consuming processed meat is carcinogenic to humans, group 1, and it causes colorectal cancer. A risk assessment determines the probability or chance of developing colorectal cancer associated with a given level or a pattern of eating processed meat. Next slide, please. Beginning first with the revised preamble, which is an overview of the updates of the monograph procedures. The preamble was published almost 2 years ago, but it has been published in practice only recently. The procedures for carrying out the monograph evaluation are described in this revised preamble, which is printed in each monograph volume. The preamble lays out the steps for participant selection in the monographs, for management of conflict of interest, for the conduct of the meeting, and for the evaluation itself. The preamble, when it was updated, it emphasized the increasing transparency and rigor of the systematic review process, as well as modernized methods for the literature searching and screening. This slide outlines all the procedures that we use at the broadest level based on the preamble, from selecting agents to selecting work grouping experts to preparing for the meeting and all the steps the secretary undertakes. And then at the heart of the monographs, which is the meeting in which the three evidence are brought together to produce summaries and evaluations of the evidence into carcinogenicity. Then at the end of the meeting that leads to the publication and a short summary that is also published, as I will explain later. So next slide, please. So how do we get nominations for evaluations? We reach out to the public and to the scientific community on a continuous basis. So nominations are open at this moment for anyone to suggest any agents that should be evaluated by the monographs. And since 2003, this has been a public call as well. However, the priority groups are convened on a 5-year basis. Selection and timing of evaluations are determined by IARC in consultation with the World Health Organization in Geneva. Next slide, please. We are undertaking engagement during the development of the individual monographs. This timeline shows the steps that are undertaken, starting about a year in advance, where we provide a preliminary list of agents, have a call for data and experts to request to attend the meeting, and also we allow requests for observer status, which is an important part of our transparency in the rules, and we post the World Health Organization Declaration of Interest form. This call for experts is closed about 8 months ahead of the meeting, and the request for observer status is closed 4 months ahead of the meeting. For transparency purposes, the list of participants is announced 2 months ahead, and the call for data is closed 1 month ahead in advance of the meeting. The meeting is held usually is face-to-face, but this year, exceptionally, it has been remoted. After the meeting, the Lancet Oncology publication is put forward between 2 and 4 weeks after the closure of the meeting. The reasoning that the working group used to reach its evaluation is summarized so that the basis for the evaluation offered is transparent. Then the publication of the book or the monograph follows about 9 months after the closure of the meeting. So next slide, please. So how are the evaluations conducted? The IARC monographs evaluations are carried out in 5 steps, 5 systematic review processes. Step 1 is to identify the relevant studies by conducting literature searches. Step 2 is to screen, select, and organize the identified studies. These 2 steps are done by IARC scientists who are called the Secretariat. Step 3 through 5 are carried out by the working group. That means the experts, the external experts. And the study quality is evaluated in step 3. Study characteristics are reported in step 4. And in step 5, and at the 8-day meeting held in Lyon, the evidence is synthesized, leading to an overall evaluation. In step 5, the evidence of carcinogenicity is first synthesized individually in each evidence stream. Next slide, please. Studies considering by the working groups are epidemiological studies on cancer in humans, experimental studies in cancer in animal models, mechanistic studies, as well as any other relevant data. key studies are identified by using systematic review principles as described in the preamble. Next slide, please. So regarding the assessment of exposure, increased attention has been put on the quality of the exposure assessment methods, and for each selected study, the exposure assessment method is summarized. Next slide, please. So what is new in this revised preamble regarding the exposure assessment? So the revised preamble reinforces the descriptive aims and call for enhanced low and middle income countries coverage, and it has a new selection on critical review of exposure methods in the epidemiological studies of cancer, and mechanism is provided. Next slide, please. So the ability of the body of the evidence to inform the working group about the carcinogenicity of the agent in studies of human cancer is related to both the quantity and the quality of the evidence. The type and quality of the exposure assessment methods are key considerations when interpreting findings for hazard identification. Next slide, please. So what's new in this revised preamble is an enhanced focus on systematic review tools, an explicit consolidation of study informativeness as well as quality, and an emphasis has been put on impact of bias and confounding. Next slide, please. So high quality experimental studies on cancer in experimental animals are considered in subgroup 3. Next slide, please. And what is new in the preamble is that studies on genetically modified animals are eligible, as explained in this slide. For example, studies including TGAC or VHA-RAS oncogene, or TRP53 or RAS-H2, as well as mice with target expression of vital genes, or humanized mice implanted with human cells normally infected by viruses. Next slide, please. Mechanistic data may provide evidence of carcinogenicity and may also help in assessing the relevance and importance of findings of cancer in experimental animal and in humans. Analysis of mechanistic data can be challenging as mechanism studies have gained in prominence, increasing in their volume, diversity, and relevance to cancer hazard evaluation, whereas studies pertinent to other streams of evidence evaluating the monographs, such as studies in humans and lifelines cancer bioassays in rodents, may only be available for a fraction of agents to which humans are currently exposed. There are many questions and many challenges in this process. So, for example, how to search systematically for relevant mechanisms, how to bring uniformity across assessments, and how to analyze the voluminous mechanistic database efficiently, how to avoid bias towards favored mechanisms. And mechanistic studies and data are identified, screened, and evaluated for quality and importance to the evaluation by using systematic review principles as described in the preamble. The working group synthesis reflects the extent of available evidence, summarizing groups of included studies with an emphasis on consistencies or differences in results within and across experimental designs. Great emphasis is given to informative mechanistic evidence from human-related studies than to that from other experimental test systems, and gaps are identified. Next slide, please. To address these challenges, the key characteristics of human carcinogens were introduced to facilitate systematic consideration of mechanistic evidence in the IARC monographs evaluation. And I listed in these slides a series of publications which summarize these key characteristics of human carcinogens. As reported in the slide, the key characteristics described by Smith et al. In 2016, such as it is genotoxic, is immunosuppressive, or it modulates receptor-mediated effects, these are based on empirical observations of the chemical and biological properties associated with human carcinogens identified by the IARC monographs program up to and including the volume 100. Next slide, please. The list of key characteristics and associated endpoints may evolve based on the experience of their application and as a new human carcinogens are identified. Key characteristics are distinct from the hallmarks of cancer, which are related to the property of cancer cells. And the key characteristics are also distinct from hypothesized mechanistic pathways, which describe the sequence of biological events postulated to occur during carcinogenesis. As such, the evaluation approach based on key characteristics avoid a narrow focus on specific pathways and hypotheses and provides a broad, holistic consideration of the mechanistic evidence. Next slide, please. So regarding the available evidence, so first, the cancer in humans. Next slide, please. The available evidence for each cancer site is classified as sufficient, limited, inadequate, or as evidence suggesting lack of carcinogenicity. The ability of the body of evidence to inform the working group about the carcinogenicity of the agent is related to both the quantity and the quality of the evidence. Next slide, please. Next, for studies of cancer in experimental animals, the evidence overall is classified into the same 4 categories. Then the mechanistic evidence is classified as strong, limited, or inadequate. Next slide, please. Strong mechanistic evidence falls into one of 3 categories. The agents may belong to a mechanistic class of agents that have already been identified as carcinogenic or probably carcinogenic to humans. Or agents may have evidence related to key characteristics of carcinogens, or the evidence might indicate that a mechanism that operates in experimental animals is not relevant to humans. Next slide, please. In the last step, the 3 different streams of evidence are brought together to indicate the overall classification of the agent into one of 4 categories. Group 1, carcinogenic to humans, and we have in this category 121 agents identified so far. Group 2A is probably carcinogenic to humans, and we have 88 substances in that group. Group 2B is possibly carcinogenic to humans, and we have 313 agents in that group. And the third group is not classifiable as to its carcinogenicity to humans, and we have in that group currently 499 agents. Next slide, please. So the 3 evidence streams are brought together to provide an overall classification of the carcinogenicity hazard of the agents. The combination of the evidence leading to these overall classifications are shown here. Next slide, please. A group 1 classification, that means carcinogenic to humans, is based on either a sufficient evidence of cancer in humans or a combination of sufficient evidence of cancer in experimental animals and strong mechanistic evidence in exposed humans. Next slide, please. A group 2A classification, that means probably carcinogenic to humans, is based on two streams of evidence, at least one of which is in humans or human cells. For example, limited evidence of cancer in humans, plus either sufficient evidence of cancer in experimental systems or strong mechanistic evidence. Another way to reach group 2A is with sufficient evidence of cancer in experimental animals and strong mechanistic evidence in human cells or tissues. Lastly, group 2A may be reached based on mechanistic class consideration if there is a structurally and toxicology similar chemical already in group 1 or group 2A. Next slide, please. A group 2B classification, that means possibly carcinogenic to humans, is based on only a single stream of evidence, either limited evidence of cancer in humans or sufficient evidence of cancer in experimental animals or strong mechanistic evidence. Next slide, please. A group 3 classification, that means not classifiable as to carcinogenicity to humans, is based on a situation where there is sufficient evidence of cancer in experimental animals, but also strong mechanistic evidence that the mechanism does not operate in humans. All other situations not listed above would lead to a group 3 evaluation. Next slide, please. This slide illustrates some of the agents that have been classified by the IARC monograph program. Tobacco smoking, alcohol consumption, radon, asbestos, outdoor air pollution are all agents that have been classified in group 1, carcinogenic to humans. Remember that this classification does not mean that all these different substances produce the same level of cancer risk for human exposure. The classification does indicate the strength of the evidence that these agents can cause cancer. Exposure to some of these agents may be outside of the personal control of the individuals, for example, air pollution, second-hand smoke, or some occupational exposures. However, some agents, individuals can make choices that will reduce their exposure to the carcinogenic risk, for example, tobacco or alcohol consumption. Through public health actions, for example, anti-tobacco campaigns, clean air regulations, or occupational exposure limits, exposure to human carcinogens may be reduced. However, IARC does not set nor explicitly recommend policy decisions regarding such exposure reductions. These actions are within the remit of other public health organizations, nationally or internationally. Next slide, please. Results of evidence synthesis and consensus evaluation are published in the Lancet Oncology after each meeting. Next slide, please. Next slide, please. So, what are the priorities recommended for IARC monographs evaluation for the period 2020 to 2024? These are the agents giving a high priority and that are placed in the inner circle. So there, I'm listing here the different, I think you went one slide too far, so go back to the previous slide, please. So these are all the agents, and you can see it's a very large amount of agents, and the closer they are to the inner circle, it was the stronger the recommendation to evaluate them sooner. So you can go to the next slide, please. So to set the priorities, streams of evidence are based on mechanistic evidence, human cancer, cancer bioassays, bioassays plus human cancer data, mechanistic plus bioassay data, mechanistic plus human cancer data, mechanistic plus human cancer plus bioassay data, and so on and so forth. And these are listed on the left part of the slide with different colors. So red for mechanistic, blue for human, and so on and so forth. The report of the advisory group, and this is this people picture here, including several colleagues from the US trained at Harvard. And this advisory group recommend priorities for the IARC monograph, and they have done their last report last year, recommended for 20 to 24. And this also, there's a publication about these priorities published in Lancet Oncology. In the following slides, I will show a list of the priorities in more detail and what are our working plans for the years to come. Next slide, please. So flame retardants and pesticides were listed as priorities recommended for IARC evaluation for the period 20 to 24. And you can see the closer to the center of the circle, the higher priority. And also we have to group them according to their effect and the experts that we can convene in one meeting. So a large number of chemicals can be evaluated in one meeting. Next slide, please. Regarding bioagents, smoking and tobacco-related products, so these were the recommended priorities for us to evaluate in the years to come. And acrolein has been evaluated recently, and we are going to do evaluations in the other substances very soon. Next slide, please. About dyes, solvents, and other chemicals. So this is the list of priorities, and I leave you a few seconds for you to browse them. And the ones with an asterisk are the ones that are already being worked with for the next volumes of the monographs. Then we can move to slide 38. Next one. These are the medical, dietary, and complex exposures that we plan to evaluate. So there are several priority here, and I think of particularly interest may be some of the pharmaceuticals which are listed here, glucocorticoids and so on and so forth, including something very interesting, which is neonatal phototherapy. Next slide, please. So these are the metals, particles, fiber, including radiofrequency fields and electromagnetic fields. So these have also been recommended for further evaluation. Next slide, please. So 3 fully remote monographs meetings have been held so far in year 2020. And it has been quite a challenge to adapt the time zones that span across the globe, because our experts come from all continents. And to establish a report and to foster frank and scientific exchange in these new conditions, because usually the experts spend 10 days basically in Lyon working intensively with each other, and now we have to do everything remotely. But these meetings, after all, went quite well, and the monograph teams now is already preparing the working program for 2021, and the next products to be evaluated will be gentian violet, leucogentian violet, malachite green, and so on. And then we have the second volume for year 2021 is in the planning and will be announced soon. So next slide, please. So it's, as I said, I don't work myself in this program, so I'm the head of the organization, and there is a very large group of colleagues working in the monographs program. In particularly, I would like to thank Dr. Mari Schubauer-Beringen, who is the head of the program, as well as Dr. Kate Guyton, who is the senior toxicologist, and all other scientists and staff members of the monographs team that have helped me to prepare the presentation. And I would also be very happy to entertain questions and comments. And also, since we are speaking to colleagues at Harvard, in particular to students, graduate and postgraduate, to invite you to eventually participate in monographs meetings, if you are interested in learning more about the process, because we do accept observers, we do accept students, and we would be very interested in having more collaboration with Harvard in this sense. So to allow us to entertain some questions and comments, maybe I should stop here. And thank you very much for your attention. And I will give the floor back to Professor Albert Hoffman for entertaining questions and comments if he judges pertinent. Thank you very much.
Well, thank you very much, Dr. Weiderpaas, for this great lecture. The process and the outcomes of the monographs, and on your plans, quite extensive plans. Indeed, we have a few, well, quite a few actually, questions in the chat, and I'll read a few for you. So thank you for allowing us to have these questions. One is of an alumnus of our Master of Science program, Dirk Pellepies, who's working now in Germany. And I read the question directly. Why does IARC not provide risk assessments of carcinogens? In Germany, we provide exposure-response relationships, pure hazard identification of exposures, such as shift work, diesel motor emissions, or formaldehyde, are of comparably little value. It is important to give guidance on safe levels or levels with minimal risk. What would you say there?
Thank you. This is a very interesting question. So the monographs program is designed to assess hazard, not risk. And the preamble publication, which I described in quite some detail, is go quite in deep in why we do that. But in short, risk is, I would say, is a political decision, more than a decision that IARC should take. Let me give a practical example. Air pollution levels. Let's compare air pollution levels in some countries across the world. They differ quite dramatically. And what's acceptable in some countries, it would be unacceptable in other countries. And who decides who is acceptable and not acceptable actually is a political question. It's a question of how much investment and money you are willing to pay or to invest in society to have clean air. So, and it's really beyond IARC's mandate to assess risk. So we voluntarily keep out of that. So we only assess the inherent property of an agent to cause cancer. And we let the national agencies in Germany, in the US, and in each country to have their own risk assessment. So they have to take decisions themselves, which is the acceptable level of exposure that they are willing to take.
Thank you. I take the liberty to go to a question of one of our faculty members, Dr. Suji Ogino. Likely many substances, he says, have different effects in different contexts. Interactions with genetic and other exposures, for example. Some molecules may be only carcinogenic with other molecules. Is there a way to address this complexity? What is your take on that?
So it's a great pleasure to hear from Dr. Ogino. So my greetings to him. Yes, this is a challenge. So we have been working with complex substances, and complex substances can be, as Dr. Ogino mentioned, mixtures of chemicals or chemicals that interact with biological process. And I give an example: air pollution is a mixture, is a complex mixture of chemicals. And there are many other examples of substances that are mixed in ways which is very hard to make a risk assessment. But we have done it, and this is within the preamble. We have incorporated in the methodology how to do that. So basically we follow the same order, and we try to find studies, if there are enough, that have the same type of enough information to allow an evaluation. So the limit is actually, it's not the methodology, is the amount of studies available to study, because we need quite a substantial number of studies to do evaluations. So the concentric circles I showed you with our priorities, they are settled based on what's available in terms of quantity of the studies to allow evaluations.
Thank you. We have a question of Dr. Juan de Cruz. Why do we have differences between IARC and EPA classification of carcinogens in water?
Not only in water, I think there are differences in also several other classifications of agents. So the methodologies differ. So our methodology is very specific, and as I described, therefore I took quite a lot of time to describe in detail, because in a way I anticipated this question. So the methodology is being slightly different. The results might be different. And also the studies included. So we have, as I show, we have this timeline. So we cut the inclusion of the studies at some point in time, and we include the studies up to that point in time. If an agency does an evaluation 1 year or 2 years before or after, the studies that will be included in their evaluation may differ, and then the results may also differ, be updated, for example.
Thank you. Then another type of question, and I may summarize it a little bit, but the question is, how do you address the problem that invited experts who published on the exposure under investigation may wish to defend their own results? Would it not be preferable to have expert groups who did not publish on this exposure? And how about industry experts? And let me add to this a comment that has been made before as well. The glyphosate discussion that emerged after the work of IARC and in the monograph. Perhaps you could address those two questions.
Okay, so the experts. So first of all, to make an expert evaluation, you need experts. You need people that really know this subject matter. So to make an evaluation with people that are not experts on the matter, I think it would be very, very challenging, and I really doubt if it would be meaningful. So I believe that experts should be involved, and this is how our methodology is based on selecting the best experts in the world in a particularly area of knowledge. Remember, we have studied over 1,000 carcinogens, thousands agents. So, and in each case, we find enough studies, so it needs to be a body of studies, and then we look very carefully who are the experts that have been publishing in that. And then we do have a very thorough process regarding conflict of interest, and we follow that very closely. So in general, or as a rule, we don't accept experts from the industry that might have a conflict of interest. So we work mostly with experts from the public sector and not having a conflict of interest. And we go quite deeply in applying that. So it's a very rigorous process. Regarding glyphosate, I have prepared a few additional slides. I don't know, Professor Hoffman, if you would have time to go through them, or if you prefer me to just summarize orally.
Well, you can show the slides. If Coppelia can share
Yes,
them with us?
okay, that would be very good. So Coppelia, if you could please go back to the slides. So if you could go back to next slide, please. So just a few words about the glyphosate, because I believe there has been quite some interest about it in the US. So next slide, please. So the evaluation was done in 2015, so in March, and we did follow the procedure as I described before. So we had the meeting announced in 2014. We had a call for data. We have a call for observer status. We have a World Health Organization conflict of interest posted of the experts. We had the all due procedure as always. We have published the data, all references are shared, and the monograph was published. So the IARC meetings, they are open and they do follow a very transparent way, and all the methods are published. And the participants, all participants had full access to the published data, and all the publications are freely downloadable. So next slide, please, Coppelia. So specifically for the glyphosate studies, there were over 1,000 studies that were identified in screening, and in particular laboratory studies where there were pure glyphosate being evaluated, as well as glyphosate formulations. And there were in particular studies in cancer in mice, in rats, and DNA damage, including genotoxicity. And there were also human studies, that mean real world exposure in relation to DNA damage, studies comparing community residents before and after spraying, and cancer in humans, in farmers and other workers. So, and in the published monograph, we have over 250 references. So next slide, please. So the studies in exposed workers, so studies in humans, the conclusion was that there was limited evidence for non-Hodgkin lymphoma. And these were based in case control studies from Sweden, Canada, US. So we all together with over 2,500 non-Hodgkin lymphoma cases that indicated increased risk, which was not explained by other pesticides, as well as cohort studies from the US in 2 states, including 92 non-Hodgkin lymphomas that show no significant increase in risk, and also a meta-analysis of which is an objective method to combine all studies indicating an increase in risk. Next slide, please. Also, there were studies in cancer in mice fed with glyphosate and indicating a positive result in 2 of 2 feeding studies for rare cancers, which are actually extremely important for assessing human risk, but they are challenging in terms to detect signals from background noise because of high statistical significance, tumors in the absence of toxicity, and evaluation which is fully in line with acceptable principles, and also the causal relationship established. So the evaluation for cancer in animal was that it was a sufficient evidence of cancer in animals. Next slide, please. So regarding damage to DNA or genotoxicity, there was there were studies available on residents in sprayed communities, and DNA and chromosome damage in blood. And it was a strong evidence for glyphosate formulations in exposed community residents, and strong evidence in glyphosate, but there are no studies in exposed humans. So in these experiments, we're using human cells, animal cells, mammal and non-mammal, and negative to bacteria. And the other studies were also using different methods. Next slide, please. So if we would look at the 10 characteristics of carcinogenic, the studies on glyphosate mainly are in category 2 and 5, so the genotoxicity and inducive or oxidative stress, and they were showed strong evidence in both. that could be applicable to humans. Next slide, please. So in summary, for the glyphosate evaluation, so the cancer in humans in non-Hodgkin lymphoma was classified as limited. In animals, it was classified as sufficient, and the studies on DNA damage and oxidative stress as strong evidence. And in combination of these three criteria, the overall evaluation was group 2A, probably carcinogenic to humans. Bert, I hope this sort of clarifies the glyphosate part.
Thank you. Thank you very much. Yes, that was very illustrative. We have a few more questions. What about microbiome research and, study of bacteria and viruses as pathogens for cancer?
This has not yet been evaluated, so it will be very interesting to evaluate, I believe, in a few years' time. But for the time being, the evidence is still accumulating, so it is not likely not to be evaluated in 2021, but later than that.
Okay, thank you. Perhaps one of the final questions. Dr. Patricia Erickson. Can the IR classification change as more information becomes available, for example, from group 1 to 2B or so?
Absolutely, yes. So this is absolutely the case. So as evidence accumulates, substances can be reevaluated. So we have this continuous nomination of substances to be evaluated, and then there is a working group, a specific working group that decides which ones should be prioritized. And then they publish this prioritization, and then we follow the order that the experts advise us. So whenever there is accumulating evidence that things have changed, we do repeat the evaluations, and the evaluations can indeed change in both senses.
Okay. Well, Dr. Weidepas, thank you very much for sharing what you do in IARC and what your colleagues in the monographs particularly do. Very, very illustrative, I think, and impressive, I may also say. But before we let you go, we will end this by, I want to show you something. And Coppelia Liebenthal will, yes, here it is. We have the official plug that will be sent to you, has already been sent to you. It will arrive soon. In appreciation for delivering the 171st Cutter Lecture on Preventive Medicine on December 11, 2020, we extend our deepest thanks to Elisabetta Weidepas, MD, PhD. Elisabetta, indeed, our deepest thanks. With that, I would like to end this 171st, am I right? Yes, Cutter Lecture, but not without thanking the members of the Cutter Committee. This group that selects the Cutter Lecture, Dr. Mir Stamfer, Dr. Caroline Buckey, Dr. Miguel Hernan, and myself. And certainly, but not before we thank those who made this Cutter Lecture possible, particularly this remote Cutter Lecture. Elisabeth, I think, may I congratulate you? It went wonderfully well, also technically. So that is, I think, a tribute, particularly also to Coppelia Liebenthal, Ji-Young Wang, Sean De Antonia, David Shelton, who is also there when we are not remote, but still here, and David Sampson. And with that, that brings us to the end. But I would not want to leave you before, of course, thanking the audience. We're an extensive audience, generally larger than we could have had in G1, the G1 auditorium in our Kresge building. We thank the audience, we thank those who posed questions, and for those in epidemiology and those also outside of epidemiology, I wish you the best of holidays. The holiday season must and hopefully be very good for you. I wish you a great start of 2021. And in that, I close this 171st Qatar lecture. Thank you very much.
Thank you.